3. Moving into session 5, we'd like to start introducing our speakers at this time, and session 5 is on the noteworthy complex generic drug approvals, orally inhaled products. Our first speaker will be Dr. Stephen Chopsky, Staff Fellow, Division of Quantitative Methods and Modeling in the Office of Research and Standards in OGD. Next, Dr. Zhen Zhu will present. Dr. Zhu is a Staff Fellow, Division of Bioequivalence 3, Office of Bioequivalence in OGD. Following Dr. Zhu, we'll welcome Dr. Feng Yuan. Dr. Yuan is a Senior Chemist in the Immediate Office, Office of Lifecycle Drug Products, NOPQ. And our final presenter will be Dr. Andrew Klerman, Senior Physician, Division of Therapeutic Performance, Office of Research and Standards in OGD. For a complete bio on each presenter today, please refer to our speaker biographies located in the same area on our website as the conference agenda. Now, please join me to welcome our first speaker in session 5, Dr. Stephen Chopsky.
Good afternoon, everyone. My name is Stephen Chopsky, and I'm a reviewer in the Division of Quantitative Methods and Modeling within the Office of Research and Standards in OGD. I'm here to talk to you today about innovative technology, particle image velocimetry, and high-speed imaging to support approval of generic orally inhaled drug products. During my talk, I will discuss the following learning objectives. The learning objectives are to describe the current thinking on regulatory significance of spray velocity for orally inhaled drug products, to describe the approach to establish bioequivalence for inhalation spray drug products, and to understand and describe the in vitro BE studies recommended in the draft product-specific guidance for inhalation spray products, specifically the spray velocity in vitro study. Within my talk, I will be discussing spray velocity characterization. Spray velocity characterization provides an additional metric for emitted spray plume characterization. There are 3 main categories of OIDPs that are the focus of generic drug user fee amendments, or GDUFA funded research. These products are metered-dose inhalers, otherwise known as MDIs, dry powder inhalers, otherwise known as DPIs, and inhalation spray inhalers. GDUFA-funded regulatory science research on spray velocity characterization indicates that spray velocity measurements have established regulatory significance for inhalation spray products. For certain MDIs and DPIs, there is potential for spray velocity measurements to support regulatory decision-making, but further research is needed. This presentation will focus on spray velocity characterization for inhalation spray products. So what is an inhalation spray product? The FDA guidance for industry on nasal spray and inhalation solution suspension and spray drug products, chemistry, manufacturing, and controls documentation recommended in July 2002 defines inhalation sprays as follows. An inhalation spray drug product consists of the formulation and the container closure system. The formulations are typically aqueous-based and by definition do not contain any propellant. Aqueous-based oral inhalation sprays must be sterile. Inhalation sprays are intended for delivery to the lungs by oral inhalation for local and/or systemic effects. The products contain therapeutically active ingredients and can also contain additional excipients. The formulation can be in unit dose or multi-dose presentations. The dose is delivered by the integral pump components of the container closure system to the lungs by oral inhalation for local and/or systemic effects. To date, there are 4 inhalation spray drug products approved on the market. According to their approved labeling, these 4 inhalation spray products utilize the Respimat soft mist inhaler device to produce a metered slow-moving aerosol cloud following actuation. There are no generic products available. The draft product-specific guidance recommendations for each inhalation spray product in the table is available on the FDA website for generic drugs. Inhalation sprays exhibit many similar features to aqueous-based solutions for nebulization, aqueous-based solution nasal sprays, and propellant-based solution MDIs. The sprays from inhalation spray products that are currently marketed have the following characteristics: aqueous drug solution droplets resembling a nebulized aerosol, a longer spray duration that is approximately 10 times that of an MDI product, and a slower moving aerosol with a velocity that is approximately 1/10 that of an MDI product. These characteristics may impact how the inhalation spray is used as well as its performance. Development of bioequivalence recommendations for inhalation spray products has been supported by GDUFA-funded research. To address the unique features of inhalation spray products, the current method being used is based on the aggregated weight of evidence approach. The weight of evidence approach is comprised of in vitro BE studies and pharmacokinetic or PK/BE studies. The in vitro BE studies are single actuation content, aerodynamic particle size distribution, spray pattern, plume geometry, priming and repriming, spray duration, and spray velocity. While most of the in vitro BE studies shown in the list have been recommended for other types of orally inhaled drug products, the spray duration and spray velocity studies are specific to inhalation spray drug products. The in vivo BE studies are a comparative PK study with fasting single-dose two-way crossover design in the general population. All strengths of the product are to be tested where applicable. It is recommended for a generic product to have formulation sameness based on the qualitative and quantitative comparison to the reference listed drug product, as well as device similarity. Each component must be satisfied for BE to be established. No comparative clinical endpoint or pharmacodynamic BE study are recommended for inhalation spray drug products, although these studies are recommended for other types of orally inhaled drug products. Spray velocity studies require some unique considerations, which we will be discussing next. The draft product-specific guidance recommendations for inhalation spray products are available on the FDA website for generic drugs. The design of the spray velocity study recommended in the product-specific guidance is as follows. The spray velocity test should be performed at the beginning and end life stages of the product. High-speed imaging, particle individual velocimetry, phase Doppler anemometry, or other suitable methods may be used to determine spray velocity. The equivalence is based on population bioequivalence, or PBE, or other statistical analysis of plume front velocity. Plume front velocity is the velocity at the front edge of the aerosol cloud. Since the velocity is dependent on the distance from the nozzle, one distance between 8 to 12 centimeters from the nozzle is recommended for this assessment. If other statistical analysis is used, it should be adequate considering the purpose of the study and scientifically justified. Full plume front velocity versus distance data should be submitted as supportive evidence for equivalent spray velocity, which may be used to confirm the appropriate distance from the nozzle used for collecting recommended plume front velocity data. The current labeling for marketed inhalation spray products states that the device uses mechanical energy to generate a slow-moving aerosol cloud of medication. Spray velocity is expected to influence drug deposition in the mouth and throat regions and in the lungs. Spray velocity may be determined by utilizing high-speed video recording, or HSV, as may be found in the literature in this study by Hottrainer et al. A high-speed camera was used to predict the plume front velocity by photographing the developing aerosol cloud using the dark field technique to illuminate the spray. These images captured the time evolution of the spray. Data were collected from the moment the spray begins to develop to the last moment when the spray is formed at the nozzle outlet. The plume tip displacement relative to the inhaler nozzle was measured for each frame using a measuring scale. Plume front velocity was determined using a curve fit of the distance versus time data and taking the time derivative of the resulting polynomial equation. The plume front velocity was determined for the 10 centimeter distance as measured from the nozzle outlet using interpolation. Spray velocity may be determined by utilizing particle individual velocimetry, or PIV, as may be found in the literature in the study by Tamura. The laser light sheet illuminates the spray. Data were collected from the moment the spray begins to develop to the last moment when a spray was formed at the nozzle outlet. The plume tip displacement relative to the inhaler nozzle was measured for each frame using the image calibration scale. Plume front velocity was determined using a curve fit of the distance versus time data and calculating the time derivatives at distances of 8 and 10 centimeters from the nozzle outlet. We will continue discussing the use of PIV to determine spray velocity using a case study. For our case study, we will be discussing the evaluation of spray velocity from tiotropium bromide inhalation sprayed metered inhalers. The FDA is currently executing a spray velocity study for tiotropium bromide inhalation metered spray as part of GDUFA-funded regulatory science research on spray velocity characterization. Plume front velocity, or the velocity at the front edge of the aerosol cloud, was assessed using PIV or HSV recordings of a tiotropium bromide inhaler. An example of the experimental setup for the PIV studies is shown in the image at right. The PIV image sequence provides images captured from the moment when the spray begins to develop to the last moment when the spray is formed at the nozzle outlet. These images are shown below. The methods for determining plume front velocity are similar to the approach of Tamura. The PIV image sequence was used to visually determine the time duration between the beginning of the spray at the nozzle outlet and when the plume tip reaches the edge of the camera frame. Software was used to track the movement of the front edge of the plume. The elapsed time and the distance traversed by the plume front is collected and plotted. The plot shows the distance traveled by the plume front as a function of time. The velocity is determined by calculating the slope of the distance versus time curve using a backward differencing scheme. Plume front velocity was determined at 6, 8, 10, 12, and 14 centimeters downstream of the nozzle outlet. To determine the plume front velocity at a specific distance, linear interpolation was used. The distance versus time curves were assessed with HSV and PIV and compared with literature data for tiotropium bromide inhalation spray metered inhaler. The distance versus time curve is consistent with the work of Trainer et al. For an inhalation spray using the Respimat inhaler. The plume front velocity measurements were consistent with findings by Tamura. A comparison cannot be made for plume front velocity data in Trainer et al. Because a tiotropium bromide inhalation spray metered inhaler was not tested in the study. The selected distances of 6, 8, 10, 12, and 14 centimeters were assessed on the velocity versus distance curve to find the most appropriate distances for assessment of spray velocity. An 8 to 12 centimeter distance range was selected due to relatively low variability in the region beyond 6 centimeters and comparison to literature data. In addition to plume front velocity data at the selected distance used for PB analysis, the draft product-specific guidance on tiotropium bromide inhalation spray also recommends prospective applicants consider submitting full plume front velocity versus distance data as additional supportive information. The design of the spray velocity study recommended in the product-specific guidance permits other methods of determining spray velocity. Any suitable method may be used provided the methods are appropriate for assessing the spray velocity and adequately sensitive in detecting spray velocity differences between the test and reference inhalation sprays. It is encouraged that prospective applicants submit a pre-ANDA product development meeting request to discuss their method development and statistical analysis plan for spray velocity study designs. We refer you to the FDA guidance for formal meetings between FDA and ANDA applicants of complex products under GDUFA. The meeting package should include details on the method development and validation, preliminary studies, if any, and a proposed statistical analysis plan. In conclusion, inhalation sprays are propellant-free, typically aqueous-based formulations containing drug products intended for delivery to the lungs by oral inhalation for local and/or systemic effects. Establishment of bioequivalence for inhalation sprays is based on the weight of evidence approach consisting of in vitro and in vivo studies. For spray velocity studies, the plume front velocity at one selected distance between 8 to 12 centimeters from the nozzle should be measured using a suitable and sensitive technique. full plume front velocity versus distance data also serve as additional supportive information. Prospective applicants are encouraged to submit a pre-ANDA product development meeting request for discussing with the agency their development program for inhalation spray products. For future work, the FDA is currently working on completing the RLD arm of the spray velocity study for tiotropium bromide inhalation metered spray. The data from this study will be published when available. Here's a challenge question for you. Plume front velocity is defined at what specific location? A. Velocity at the front edge of the aerosol cloud. B. Average velocity along the center line of the fully developed plume profile. C. Maximum velocity measured at a central location downstream from the inhaler nozzle. Or D. Average velocity measured only at the inhaler nozzle. The correct answer is A. Velocity at the front edge of the aerosol cloud. Here's a challenge question for you. Plume front velocity may be determined from which of the following data sets? A. PIV data. B. High-speed imaging data. C. Both A and B. Or D. Neither A or B. The correct answer is C. Both A and B. For our call to action, if you have questions regarding a spray velocity study, consider requesting a pre-ANDA product development meeting. I would like to thank my colleagues who helped contribute to this slide presentation. This is a list of applicable resources.
Thank you. I'm glad to be here and talk to you about the first generic drug approval, and this is a case of Budesonide and Formoterol fumarate dihydrate inhalation aerosols, a generic version of Symbicort pressurized metered-dose inhaler from the bioequivalence perspective. First, I'll introduce you some background information, give you an overview of locally acting orally inhaled or nasal drug product, OINDPs, and FDA's weight of evidence approach and equivalence assessment for one category of OINDPs, a suspension metered-dose inhaler. Then I'll discuss this case study that used alternative approach, a sensitivity analysis to support a non-quantitative or non-Q2 formulation of generic MDI product. Following this presentation, I hope you will be familiar with the bioequivalence studies recommended in the product-specific guidance for this MDI product. The OINDPs on the market can be classified into 5 categories: MDIs, DPIs, nebulizers, soft mist inhalers, and nasal spray or nasal aerosols. Each category has its distinctive features, and they are complex drug and device combination products. Their aerosol performance depends on the interaction between the formulations and delivery devices. In turn, a successful drug administration depends on the user interface and patient's inhalation maneuver. Most OINDPs are locally acting drugs for the treatment, for example, asthma and COPD. Locally acting means that the target efficacy does not rely on systemic circulation. Based on the drug mechanism of action, the OINDPs can also be roughly classified as beta-2 agonist, anticholinergics, corticosteroids, and a combination of either beta-2 agonist with anticholinergics or corticosteroids. Here, we will focus on MDIs. Among them, MDIs are drug formulations either suspended or dissolved in an inert propellant, typically hydrofluoroalkane, HFA, and sealed in a canister. MDIs have a metering chamber and an actuation mechanism. When actuated, the vapor pressure forces a pre-metered dose out of the actuator nozzle to form a respirable plume. Then an actuation inspiration coordination is required to get correct aerosol dose into the respiratory tract or lungs. Because there are numerous uncertainties in the complex drug-device combination products, including MDIs, we recommend aggregate weight of evidence approach for the generic drug bioequivalence. They include 4 coherent elements: equivalent in vitro performance testing, including single actuation content, aerodynamic particle size distribution study, spirit pattern, plume geometry study, priming and repriming studies, and equivalent systemic exposure based on in vivo pharmacokinetic data, and equivalent local delivery based on a clinical endpoint study or alternative approach, and formulation sameness and device similarity between proposed test product and reference product, including device component sameness and formulation qualitative and quantitative or Q1, Q2 sameness. These 4 elements are evidence to support both equivalent efficacy and equivalent safety. None of them, when standing alone, would provide adequate assurance of the equivalence in both safety and efficacy. With these background information in mind, I will discuss a special case on approval of a non-Q2 formulation generic version of Symbicort. The reference listed drug, Symbicort, was approved in July 2006. This suspension MDI is indicated for the treatment of asthma in patients 6 years of age and older, and maintenance treatment for COPD. The ROD has 2 strengths, 80 or 160 microgram of budesonide per actuation, and a fixed strength of 4.5 microgram of Formoterol fumarate per actuation. The product-specific guidance for this drug product was published in 2015. The agency recommends 5 in vitro studies on both strengths, including SAC, APSD, spirit pattern, plume geometry, priming and repriming, and in vivo PK studies on the fasting condition on both strengths, and 1 comparative clinical endpoint study on the lowest strength in asthma patients. In addition, the formulation sameness and device similarity is also recommended. However, during the review of this application, it was found that the proposed formulation was Q1 the same, but not Q2 the same as the ROD, because one of the excipients, here we designate it as excipient A, has percent difference greater than 5% between test and reference product. The excipient A did not exceed previously approved drug product intended for inhalation. This means there's no safety concern. The applicant submitted all 5 in vitro studies, in vivo PK studies, and comparative clinical endpoint studies as recommended in the PSG. In order to support non-Q2 formulation, the applicant conducted additional in vitro sensitivity studies to evaluate the impact of varying amount of excipient A on aerosol performance. The study report demonstrated the formulation understanding based on pharmaceutical development data and elaborate on how the proposed test formulation was selected. The sensitivity studies include the above-mentioned 5 in vitro studies, but conducted on 3 levels of the excipient A: lower than test, equal to, and higher than ROD. These 5 studies were designed to be 3 batches with 3 levels of excipient A for each strength, and 10 samples in each batch. PBE analysis were conducted to evaluate the equivalence between test and reference. In addition, 2 suspension formulation characterization, including morphology and stability studies, were also conducted. The result of all in vitro studies and suspension formulation characterizations indicated that varying amount of excipient A did not affect the aerosol performance. The morphology characterization and suspension stability show similar results among test formulation with varying amount of excipient A and ROD. This slide shows a hypothetical data set for SAC results. The study was conducted at the begin, middle, and end life stages, and the mean delivered amount was presented as a percent label claim. You can see that the mean delivered budesonide and Formoterol fumarate from the test product at 3 life stages was comparable to that of the RD product, and the mean delivered amount was roughly within 95 to 105% of the percent label claim in all cases. The study passed PB analysis. Similar outcomes were also observed for priming and repriming after non-use and dropping. Eventually, the first generic of Symbicort to treat asthma and COPD was approved on March 15, 2022. So, a non-Q2 application may be submitted to FDA for MDI, and this generalized to all OINDPs. However, sufficient data and comprehensive information are needed to justify the impact of the formulation differences on bioequivalence and safety. For example, if SAC study shows consistently lower percent label claim for the test product than the RD product, as the graph show here, it would likely fail to meet the PBE criteria. Then, an investigation should be conducted to determine whether this lower delivered dose is caused by the differences of the formulations between the test and the RD products, after excluding numerous confounding factors that may also affect aerosol performance. In summary, FDA recommended the weight of evidence approach to evaluate bioequivalence of locally acting OINDPs. A case study for a non-Q2 MDI application is briefly touched. Typically, a Q1, Q2 same formulation is recommended. Scientific justification with appropriate data and scientific explanation are recommended for the development of non-Q2 formulations. FDA recommends the applicant to follow product-specific guidance for efficient generic OINDP product review. If the applicant wants to use novel B approach, for example, non-Q2 formulations, in this case, FDA provides multiple communication channels, such as control correspondences, product development meeting, pre-submission meetings, mid-cycle review meeting, and enhanced mid-cycle review meetings, and post-complete response scientific meetings at various stages to address applicants' questions. We strongly encourage early communication with FDA, which could help to clarify the FDA's expectation for prospective applications early in the product development and assisting in submitting a comprehensive application. I'd like to thank you for the opportunity to present this talk to you. Special thanks to our Office of Bioequivalence Director, Dr. Patharoy, Division of Bioequivalence 3 Director, Dr. April Brady, Deputy Director, Dr. Kiran, Dr. Li Gong, and my colleagues and inhalation experts from different disciplines in the FDA, including Dr. Fan Yuan from the Office of Pharmaceutical Quality, Dr. Brian Newman, and Dr. Elizabeth Besky from Office of Research and Standards, and Dr. Channing Guo from the Office of Testing and Research. Poll question number 1. What are included in the FDA's weight of evidence approach for orally inhaled or nasal drug products? A. Equivalent in vitro performance. B. Equivalent systemic exposure. C. Equivalent local delivery. D. Formulation and device sameness. E. All the above. Take a second to consider which answer you think is correct. If you choose E, all of the above, that's the correct answer. Poll question number 2. How many types of OINDPs are available on the market? A. Dry powder inhaler. B. Pressurized metered-dose inhaler. C. Nebulizer. D. Soft mist inhaler. E. Nasal spray, nasal aerosol. F. All the above. Take a second to consider which answer you think is correct. If you choose F or the above, that's the correct answer. That concludes my presentation. I will be back shortly to answer questions. Thank you for your attention.
Good morning, everyone. I'm Feng Yuan, a senior chemist in the Office of Life Cycle Drug Products under the Office of Pharmaceutical Quality. Dr. Zhen Xu just went through the agency's bioequivalence approach for metered dose inhaler and took the first approved generic version of Symbicort for a case study. Let's switch gears and discuss some recommended considerations from quality perspective. Speaking of quality, everyone deserves confidence in their next dose of medicine. And our day-to-day work in the Office of Pharmaceutical Quality is to ensure every single marketed pharmaceutical product's quality, so to assure the availability, safety, and efficacy of every dose taken by our patients. In this 15-minute presentation, I will discuss potentially overlooked issues in formulation development of generic suspension-based MDI products, and highlight a few product characterization studies helpful to verify product design providing consistent delivered dose, or identify the risk in product design causing inconsistent delivered dose. Budesonide and Formoterol Fumarate Dihydrate Inhalation Aerosol has two strengths, and the reference product Symbicort was approved in 2006, and the first approved generic version, Brina, was approved in 2022. A few weeks ago, Viatris just announced the launch of the product. It is a suspension formulation consisting of two micronized drug substances co-suspended in the Propionate-based formulation. The formulation also contains a small amount of suspending agent and lubricant. And this table here provides the metered dose by the valve and the delivered dose from an actuator for both strengths. The co-suspension-based MDI product is a perfect representative of the challenging dosage form that I will discuss in this presentation. This is a cross-sectional diagram of a metered dose inhaler, and the canister contains the bulk formulation, where the concentration of the active ingredient is the assay of the product, and the total quantity of the bulk formulation filled inside the canister is the net content. And the valve outline is in green color. And in the middle of the valve, the small chamber here is the valve metering chamber. The size of the metering chamber determines the amount of the bulk formulation metered by each actuation, where the quantity of the active ingredient contained in the metered formulation is the metered dose. And the actuator outline is with the brownish red color. And at the bottom of the actuator, the extended part is the actuator mouthpiece. And inside the actuator here, the little space here is the actuator sump, which provides the moving chamber or moving space for the valve stem during an actuation. And close to the end of the actuator sump, the tiny opening here is the actuator orifice. So when a metered dose inhaler is in rest position, the metering chamber is open towards the formulation reservoir, and upon actuation, the metering chamber became closed towards the formulation reservoir and open to the atmosphere, which leads to the expansion of the metered formulation moving through the valve stem and subsequent atomization from the actuator orifice. So the quantity of the active ingredient discharged from the mouthpiece is the delivered dose, and the target delivered dose defines the strength of the product. So one of the major challenges in developing suspension-based MDI formulation is to maintain the consistent delivery dose through unit life over shelf life. And the CQAs, including RC, net content, metered dose, all can impact the delivery dose uniformity. Here's an example of typical generic MDI product composition table. And the composition table captures a number of CQAs we just discussed. The quantity of the active ingredient is the target delivery dose, and also the strength of the product, which is always same as the RODs. And the quantity of the active ingredient metered by the valve is the metered dose, and the total quantity of the formulation metered by the valve is the valve delivery and also called the shot weight. The metered dose and valve delivery may or may not be same as the RODs because they're also impacted by the size of the valve metering chamber and the density of the proposed formulation. And the metered dose is also impacted by the RC, or the canister concentration of the active ingredient. And the total quantity of the formulation filled in the canister is the net content. And net content also may not be same as the RODs because it's impacted by the size of the selected canister in the product design. The tail-off characterization studies should be used to justify the proposed minimum net content. And all of these 4 CQAs are specified in product label, but the quantitative values in the rest of the cells in this table are not public available information. So during formulation development, one of the big challenges is to determine the target canister concentration of active ingredients, or the RC of MDI. And the target canister concentration of inactive ingredients are subject to Q1, Q2 sameness evaluation for generic product. During formulation development for MDI product, most generic applicants choose to develop a formulation that is Q1, Q2 same as the ROD. But in the case where the proposed formulation is not Q1, Q2 same as the ROD, then additional supporting data will be needed to demonstrate that the deviation does not impact the product quality and performance. You may refer to Dr. Zhenshu's presentation regarding the required supporting data from bioequivalence perspective, and from the quality perspective, the type and amount of supporting data depends on the non-Q1, Q2 same inactive ingredients function in the formulation. I will give a brief example when we discuss the product characterization studies. The target canister concentration of the active ingredient is determined based on the reverse engineering study results from the ROD formulation. Same as other dosage forms, the agency discouraged use of overage as per ICH Q8 guidance. But if the development study suggests that an overage is necessary, then applicants need to provide adequate justification and supporting data. For example, unavoidable drug loss during manufacturing process that may compromise the delivery dose uniformity. But here, I also would like to recommend our applicants carefully investigate the potential drug loss due to drug deposition on device constituent parts during use. If you recall, when we discussed the operating mechanism of MDI, upon its actuation, the formulation is metered by the metering chamber and then passing through the valve stem, actuator sump, actuator orifice, and mouthpiece before inhaled by the patient. So the drug particles may deposit at the inner surface of those device constituent parts, and the level of deposition vary from dose to dose through the unit life, usually higher at the beginning of the unit life because all the surfaces are clean, not occupied by the drug particle residue. And depending on the geometric dimension, materials of construction, and coating treatment of the selected device constituent parts, the level of deposition may vary as well from product to product. We're halfway through this presentation. Let's take our first challenge question. Which of the following critical quality attributes can impact the delivered dose uniformity of MDI products? A. Assay, B. Leachables, C. Net content, and D. Metered dose. The correct answer is assay, net content, and metered dose. The 2018 Draft Quality Guidance recommends a list of product characterization studies for the purpose of supporting robustness of to-be-marketed products' performance and labeling statements. We recommend applicants to conduct these studies using at least 3 exhibit batches that represent the to-be-marketed product, and you may refer to the draft guidance for additional details on the purpose and recommended design for individual study. But here, I'd like to emphasize that these product characterization studies can be more than a tool verifying the final or close to final design of your proposed product. And I will highlight a few characterization studies as examples to further elaborate how we can leverage them to identify the risk in product design at early stage of your development program. So drug deposition, mouthpiece, and or accessories characterization study in conjugation with cleaning instruction study can be helpful to assess the trending of delivered dose through in-use periods due to drug deposition on the internal and external surfaces of individual device constituent parts. To serve this purpose, we strongly recommend applicants to assess the trend of drug deposition, following the label-instructed cleaning intervals through unit life, and include all critical device constituent parts involved in the flow pathway of the metered drug formulation during the actuation. And for the cleaning instruction study, we recommend applicants to strictly follow the cleaning intervals through unit life and cleaning procedure described in the label. And based on the study results, carefully select critical device constituent parts and optimize the product formulation to minimize the impact of drug deposition on device constituent parts on the delivered dose uniformity through unit life. The profiling of actuations near device exhaustion study, also called tail-off study. The study can be used to justify the proposed minimum net content of the product to ensure consistent and adequate product performance through the label claimed number of doses. And the effect of storage and shaking study is to demonstrate the adequate product performance according to the label instructed shaking procedure before its actuation. And in addition, if an applicant proposed a generic formulation contains surfactants not Q1, Q2 same as the ROD, you may use the study results as part of your justification for the deviation in the proposed formulation. Now, it's time to take our second challenge question. Which of the following statements regarding product characterization studies are correct? A, to support labeling instructions, and B, to verify robustness of product design. C, one-time studies never needs to repeat. D, either using development badges or exhibit badges. The correct answer is A and B. It's not routine studies. However, if you make changes to your product design, you may need to repeat the product characterization studies depending on the level of the proposed changes. Here's a list of the related guidance and compendial standards for your reference. Take-home messages. To minimize the risk of inconsistent delivered dose during the life over shelf life of generic suspension-based MDI products, we recommend applicants cautiously select device constituent parts to minimize the drug particles built up during use, and do not overlook potential impact of drug deposition in device constituent parts on the delivered dose uniformity. And we encourage applicants to leverage certain product characterization studies to quickly identify the risk in product design that causing inconsistent delivered dose and optimize the product design at early phase of development if possible. That brings us to the end of my presentation, and thank you all for your attention. Let's reconnect during the panel discussion.
Hello, my name is Andrew Clareman, and I am a senior physician in the Division of Therapeutic Performance 1 within the Office of Generic Drugs. Today I'm going to discuss the post-approval impact of generic Fluticasone propionate and Salmeterol inhalation powder. The learning objectives for my talk include describing the high costs of inhalers in the United States and the effect on patient outcomes, identifying the difference between an approved generic through the ANDA pathway and an authorized generic, and describing the post-market impact of ANDA-approved generics and authorized generic inhalers. U.S. per capita prescription spending is higher than comparable countries. As can be seen in this figure from a Peterson KFF study, comparable countries have spent significantly less money on prescription drugs since 2004, and the difference appears to be accelerating. Looking specifically at inhalers, these are a substantial cost to the U.S. healthcare system. Between 2012 and 2018, Medicaid spent over $26 billion on inhalers, and this annual spending has increased over $2 billion, or 120%, during this time. Inhaled corticosteroid long-acting beta agonist, or ICS-LABA, combination inhalers represented 53% of that spending and increased over 600%. Finally, almost 7% of the $168 billion spent by Medicare Part D in 2018 went toward inhalers for asthma or COPD. In U.S. patients who rely on inhalers, cost barriers are associated with uncontrolled asthma in both adults and children, at least one asthma-related hospitalization in elderly patients, and in COPD patients, a higher likelihood of abandoning inhaler therapy. The FDA conducted an investigation looking at the impact of generic inhaler approval on uptake and costs. This investigation included looking at Wixela Inhub, a generic dry powder inhaler that references Advair Diskus and was approved in January 2019 through submission of an ANDA. Additionally, an authorized generic of Advair Diskus entered the market in February 2019. FDA investigators published this research that investigated the medication cost savings as well as the utilization of generic ICS/LABA products in the United States using IQVIA data. IQVIA provides information about invoice transaction pricing of pharmaceutical sales as well as dispensing. In the study, the investigators hypothesized that introduction of the first approved generic would lead to cost savings compared to an authorized generic. Let's take a moment to look at the difference between authorized generics and approved generics. Authorized generics are identical to a brand name drug. They are marketed under the same new drug application as the brand name, but do not display the brand name on the product. Comparatively, approved generics may have differences from the brand name drug, but are required to demonstrate bioequivalence through an ANDA submission. So how do authorized generics impact the marketplace? In 2011, the US FTC issued a report that found that authorized generics impact the marketplace in several ways. This included that authorized generic competition during the 180-day exclusivity period for an approved generic led to somewhat modestly lower prices of 4 to 8%. However, they also found that authorized generics have a substantial negative effect on the revenues of competing generic firms. Finally, they found that brand companies may use the threat of issuing authorized generics to prompt generic companies to delay their products, and this may lead to limited market competition and higher prices for consumers. Moving back to the FDA's investigation, they looked at a few key products within this drug class. First, they looked at the approved generic referencing Advair Diskus, which was introduced in 2019 following ANDA approval. And around the same time, in February 2019, an authorized generic of Advair Diskus was also introduced. In January of 2020, an authorized generic of Symbicort was introduced to the marketplace, providing a useful comparison. Let's first look at the wholesale units of ICS/LABAs between 2015 and 2020, which was the FDA's study period. This graph shows the wholesale units over time, with the red line on top representing the total units. The first dotted line represents when the Advair generics were introduced in early 2019, and the second dotted line represents when the Symbicort authorized generic was introduced. Now let's take a closer look at the time before generic approval. This graph shows the wholesale units between 2015 and 2018 prior to the release of the generic Symbicort and Advair products. The red line represents the total units of the ICS/LABA class, which you can see is gradually increasing over this time. The Advair brand name product is gradually decreasing, and this is likely driven by a gradual increase in the uptake of a competitor brand name product, Bioequivalence. Now let's look at what happened after generic approval. This graph shows the wholesale units between 2019 and 2020 for the Advair Diskus and its associated generics. As you can see, in early 2019, when the generics were approved, there was a rapid decrease in the wholesale units of Advair Diskus brand name, while there were increases in the wholesale units of the authorized generic, as well as the approved generic. Notably, the total wholesale units stayed relatively stable. Similarly, looking at the Symbicort brand name as well as authorized generic, the total units stayed relatively stable, but in early 2020, when the authorized generic was released, you saw a decrease in the wholesale units of the brand name and a corresponding increase in the authorized generic. Now let's look at cost. Here's a graph of the wholesale cost of ICS/LABAs between 2015 and 2020. Again, the first dotted line is when the Advair generics were introduced, and the second dotted line is when the Symbicort authorized generic was introduced. Again, we're going to look at the time period before generic approval. So this graph shows wholesale costs between 2015 and 2018. As you can see, the total wholesale cost was increasing relatively quickly. However, the wholesale cost of Advair brand name was decreasing. So the increase in wholesale costs was likely driven primarily by the uptake of the competitor brand name Bioequivalence, as well as Symbicort to a lesser extent. Now let's look at after generic approval. Here's wholesale cost of Advair Diskus and its corresponding generics between 2018 and 2020. As you can see, in early 2019, there was a rapid decrease in the total wholesale cost of this product family. This was primarily driven by a rapid decrease in the wholesale cost of Advair Diskus brand name, with a corresponding small increase in the wholesale cost of the authorized generic as well as the approved generic. Similarly, we look at the wholesale cost for the Symbicort family of products. In early 2020, when the authorized generic was introduced, we see a corresponding decrease in the wholesale cost of the brand name Symbicort, along with an increase in the wholesale cost of the authorized generic. However, there is not much change in the total wholesale cost for this product family. Now that we've looked at wholesale units and cost independently, let's put the data together. This graph shows the wholesale units of the Advair Diskus family of products. As we noted before, the total wholesale units stayed relatively stable. However, we can see that beginning in early 2019, there was a rapid decrease in the total wholesale cost as seen on the red line in the graph on the right. Therefore, despite relatively stable units being sold, there was a significant decrease in the total amount being paid for these products. Now for comparison, let's look at the Symbicort family of products. From 2018 through 2020, the wholesale units of the Symbicort family stayed relatively stable. And again, in early 2020, with the introduction of the authorized generic, there was a small decrease in the wholesale units of the Symbicort brand name. Looking at cost, the wholesale cost through the same time period stayed relatively stable. And despite the introduction of the authorized generic, there was no corresponding decrease in the total wholesale cost like we saw for the Advair family of products. Now let's put some numbers to the cost savings associated with generic Advair approval. The total cost for Advair and its generics decreased from $337 million in January 2019 to $233 million in January 2020, despite stable wholesale units. A similar corresponding decrease did not occur for Symbicort, despite the release of an authorized generic. The average unit cost for the approved generic of Advair was $115 compared to $169 for the authorized generic and $334 for the brand name. To put these cost savings into a more real-world context, let's look at GoodRx prices for zip code 20903, which is where the FDA headquarters is located in White Oak, Maryland. This was accessed on June 27, 2023. As you can see on the left, for the Advair approved generic, Wixela Inhub, the lowest price available was around $75. By comparison, the Advair Diskus brand name for the same dose, the lowest price was about $385. To summarize, the US spends a lot of money on inhalers, and high inhaler costs are associated with worse patient outcomes. The FDA investigation described in this talk found reduced costs with the ANDA approval of a generic referencing Advair. However, introduction of an authorized generic for Symbicort had limited impact on costs compared to the release of the approved generic for Advair. Now let's move to the challenge questions. Challenge question 1. According to a CDC study, U.S. asthma patients 65 years of age and older who had at least one asthma-related hospitalization in the past year were more likely to report A. Pets in the bedroom, B. Cost barriers to obtaining medication, C. Secondhand smoke exposure, or D. Obesity. The answer is B, cost barriers to obtaining medication. Challenge question number 2. According to an FDA investigation, introduction of an approved generic ICS/LABA through the ANDA pathway resulted in significant cost savings, whereas introduction of a blank did not. A, nebulized formulation. B, companion app to monitor inhaler adherence. C, authorized generic. Or D, leukotriene receptor antagonist. The answer is C, authorized generic. Here's a list of my sources for reference. If you have any questions, please submit them for the question and answer session that will follow. As a final thought, I want to leave you with this quote from one of my OGD colleagues. On behalf of OGD, I want to thank the audience and our stakeholders for their hard work bringing safe and effective generics to the marketplace. Thank you.
Thank you all for the great presentations. As another reminder, if you haven't had a chance to enter your questions into the Q&A chat pod, please do so now. In addition to our presenters, we welcome the following panelists: Dr. Srinivas Behera, Chemist, Division of Immediate and Modified Release Products 3 and OPQ; Dr. Tien Ma, Senior Staff Fellow, Division of Bioequivalence 1, Office of Bioequivalence and OGD; and Dr. Elizabeth Bilski, Senior Pharmacologist, Office of Research and Standards, and OGD. Looks like we have some questions coming in right now. The first couple of questions will be directed to Dr. Stephen Chopsky, and here is the first question for Dr. Chopsky. Why is the current thinking on regulatory significance of spray velocity for orally inhaled drug products limited to only the plume front velocity rather than the temporal and spatial development of the spray plume velocity?
Yes, thank you, Ray. That's a very good question. So, plume front velocity is the velocity at the tip of the plume, which I discussed in my talk. So, plume front velocity provides a more appropriate metric for velocity with minimal variability as compared to other velocity measurements of the plume. With regard to temporal and spatial development of the plume, there can be a lot of variability in the internal, uh, basically the internal velocities of the plume. If you were to look at local velocities, the velocity at the tip of the plume provides a very regular decline in velocity and can be effectively measured. Thank you.
Thank you for responding to that question. We've got a couple more questions for Dr. Chopsky, and here is the next question. Can you elaborate on what is the role for spray velocity measurements for demonstrating bioequivalence of meter dose inhalers?
Yes, thank you, Ray. That's a very good question. So the product-specific guidance documents, or PSGs, for all solution-based MDI products provide for an alternative bioequivalence approach to the comparative clinical endpoint study. So these additional supportive in vitro studies may include studies that support the characterization of emitted aerosol sprays with respect to velocity profiles and also evaporation rates, which would include spray velocity measurements. So I would note that spray velocity measurements, if they're used in an alternative BE approach, would be best used as in a supporting role rather than as pivotal evidence for establishing BE. I'd also like to point out that the Center for Complex Generics actually conducted a workshop back in April, so earlier this year, and highlighted the use of spray velocity measurements in a supportive role for BE. Um, if anyone's interested, you can find the proceedings for the workshop on the CRG website. Thank you.
Thank you for responding to that question, Dr. Chopsky. And we have one more question for you in this round, and here's the question. What is the purpose of the FDA working on the reference listed drug arm of the spray velocity study as described in the product specific guidances for all inhalation spray products?
Yes, thank you for the question, Ray. So the FDA is working to produce a spray velocity reference data set based on the RLD product for spray inhalation spray products. The study results may provide a very useful reference for potential ANDA applicants who are considering conducting a spray velocity study. Thank you.
Thank you for responding to that group of questions. Moving on to our next panelist, we've got a few questions that just came in for Dr. Zen Zhu. And here's the first question for Dr. Zhu. What should be submitted to meet the standards for the proposed non-Q2 formulation product? I'm sorry, Dr. Zhu, we're not able to hear you at this time. We're going to have our tech team check in with you and check your volume. We're going to move on to our next panelist, Dr. Fang Yuan. And here is the question for Dr. Fang Yuan. Does the agency expect that the product characterization studies to be performed on at least 3 batches of each strength or a total of 3 batches across the proposed strengths?
Thank you. This is a great question. So it's very common question that we encounter during the pre-NDA phase as well. So in general, we do expect that the applicant to submit product characterization studies on at least 3 batches of each strength. The reason is that we recommend at least 3 batches is to evaluate the batch-to-batch variation and the difference in strengths between strengths may the level of the variability across strengths may not be the same, and the batch-to-batch variability from the product components may have different level of impact on the end product batches. Thank you.
Thank you for responding to that question. We have another question that came in for Dr. Yuan, and here's the question. If changes have been made to the product design after all product characterization studies have been completed, does the applicant need to repeat all product characterization studies?
Thank you. Great question. So it's there is no and there is a unique rule for that. And in general, we do expect to recommend the applicant to submit a risk assessment for the changes and also and justification for the selected product characterization studies if they do not wish to conduct all product characterization studies. So it really depends on the level of changes. Thank you.
Thank you, Dr. Yuan. We have one more question for you in this round, and here is the question. Where is the ANDA submission? Where in the ANDA submission should we include the device constituent parts, characterization studies, and cleaning instruction study?
Thank you. So I'm not sure what the device constituent parts characterization studies meant for. If you're referring to the product characterization studies, then we do in general we recommend applicants to submit the product characterization studies, including cleaning instruction study in P2. But if you're talking about the information and related functionality studies on device constituent parts, then we recommend applicant to submit such information in P7. Thank you.
Thank you, Dr. Yuan, for responding to that group of questions. We're going to check back in with Dr. Zhen Zhu to see if we have connectivity with her mic. And here is the next question for Dr. Zhu. What should be submitted to meet these standards for the proposed non-Q2 formulation product?
Thank you for the question. So if a sponsor is interested in submitting a non-Q2 formulation, the sponsor should first provide an explanation why a Q1, Q2 formulation cannot be used and what is the rationale for selecting a non-Q2 formulation. Then a sponsor should demonstrate that non-Q2 formulation has no safety concern. And then after that, the sponsor should provide pharmaceutical development data, including in vitro testing of multiple drug to excipient ratio that encompass combinations below and above the ratios used in the TNR product. So this is to ensure that non-Q2 formulation does not impact the drug product performance. Thank you.
Thank you, Dr. Zhu. We hear you loud and clear. We do have a few more questions for you in this round, and here is the next question. Does the non-Q2 generic drug development strategy apply to all and orally inhaled drug products? And how about nasal non-Q2 generic drug products?
Yeah, thank you for the question. It depends. Orally inhaled products are complex and they have vast differences in formulation device technology. So they may be passive inhalers that are highly dependent on the inspiration maneuver or contain a propellant that requires hand-lung coordination. So the inhalation products have different indications that may target the locally or systemically. Or there are new formulation technologies used in getting respirable particles or need to be considered, and they will be evaluated case by case. So scientific justifications with appropriate data and explanations are recommended for the development of non-Q2 formulation, for example, those MDIs or DPI products. On the other hand, for the nasal product, for example, solution nasal product, the product-specific guidance generally have two options. The bioequivalence can be established from in vitro or in vivo options. If they are Q1, Q2, they can choose in vitro options, and typically the SAC, priming, repriming, DSD, small particle by cascade impaction, spray pattern, and plume geometry studies. If non-Q2, an in vivo PK study is generally recommended to establish bioequivalence. Thank you.
Thank you, Dr. Zhu. We do have one more question in this round, and here's the question. Why does the applicant want to overcome the hurdles and develop a non-Q2 generic drug while the easier Q1, Q2 route is accessible?
Yeah, that's a good question. So following Q1, Q2 route based on the PSG recommendation, it should be straightforward, but there might be some issues like patent issues or some unknown issues. So that drives the applicant to choose non-Q2 route. However, applying a non-Q2 generic drug product would face many more scientific and regulatory challenges for sure. This is why it's important to communicate with the agency earlier, for example, through control correspondences or pre-enda meetings to discuss the matter like those proposed non-Q2 formulation in advance. Thank you.
Thank you for responding to that group of questions. Moving on to our next panelist. A few questions came in for Dr. Andrew Klurman, and here's the first question for Dr. Klurman. Do you have any data on how the marketplace has evolved since 2020 when the FDA study was completed?
Thank you for that question. So since the study was completed in 2020, we haven't done any formal analyses, but from what we can tell, the trends that we saw in the FDA study have stayed relatively stable. Thank you.
Thank you for responding to that question, Dr. Klurman. We've got a couple more questions, and here's the next question. Do you have any data on how the marketplace, whoops, sorry. Do you, what, sorry, I read that question. Go back to the first question just came in. What do you anticipate the impact of a second, third, etc. generic approval would be on the cost and uptake within a product family?
Thank you for that question. So what we anticipate is for subsequent generic approvals to continue to lower the cost of that product family. We do have data, and I believe there's a FDA publication from a few years ago that shows that we have increasing cost savings with second, third, and fourth generics compared to just that first generic. So within the past few years, we've had some additional approvals, and we intend to continue this research to continue to monitor the cost impact and the uptake impact that these generic approvals are having. Thank you.
Thank you for responding to that question. We have one more question for Dr. Klurman, and it is the following. How difficult would it be to do a similar cost impact analysis for other generics?
Great question. So similar to the last question, I think it's not necessarily a matter of difficulty. I think we have the data. It's just a matter of having additional products to do the research on. And now that we've had a few additional approvals of various orally inhaled drug products over the past few years, we do intend to continue this research and evaluate the continued cost savings. Thank you.
Thank you for responding to that group of questions. Moving on to our next panelist, we've got a couple questions that came in for Dr. Srinivas Behara. And here is the first question for Dr. Behara. For an A&D submission, is it acceptable to manufacture one exhibit batch at commercial scale and the remaining 2 exhibit batches of the product to be manufactured at 1/3 of the proposed commercial scale?
Good morning, everyone. That's a very good question. 3 batches of drug product to be manufactured from 3 discrete lots of drug substance and critical excipients, and also device constituent part components. Among the 3 batches, at least 1 batch should be manufactured at the proposed commercial scale, and the other 2 batches at least at 1/3 of the commercial scale. All the batches of the drug products to be fully filled. For example, if we are talking about soft mist inhalers, drug product to be fully filled into cartridges. Therefore, the approach you have appears to be reasonable. If applicants have different strategies, we recommend requesting a pre-ANDA product development meeting. Hope this clarifies your question. Thank you.
Thank you, Dr. Behara. We do have one other question that just came in for you, and here is the question. From a chemistry standpoint, what are the resources for the product development of inhalation spray products?
That's another good question. The resources for the development of inhalation spray products would be MDI, DPI products quality considerations, which is April 2018 draft guidance. Product specific guidance, if any, I would say USP 5, USP 601, and nasal spray and inhalation solution suspension and spray drug products CMC documentation, which is July 2002 guidance. If you have any specific development question, we highly encourage you to request a pre-ANDA development meeting. Thank you.
Thank you for responding to both of those questions. Moving on to our next panelist. Got a few questions that just came in for Dr. Tian Ma. And here's the first question for Dr. Ma. what studies should be submitted to support the non-Q2 formulation for each of the solution meter dosed inhaler, suspension meter dose inhaler, dry powder inhaler, and soft mist inhaler?
Thank you for the question. So actually, Dr. Zhang Xu answered a very similar question earlier, and he provided the answer very nicely. So here, I just want to emphasize that we still highly recommend Q1, Q2 formulations to be provided. But if it cannot be accomplished and you have to use a non-Q2 formulation, we recommend you to communicate to the agency earlier so that to have a better understanding and discussion about what studies or information is needed for your submission. And this can be done through the control correspondence, product development meetings during the pre-ANDA phase, and also meetings such as the mid-cycle review meetings or enhanced mid-cycle review meetings or post-CR scientific meetings. Thank you.
Thank you for responding to that question. We do have one more question from Dr. Ma. Here's the question. If the RLD product changes after the completion of the bioequivalence studies, do we need to repeat all BE studies?
Thank you for the question. This is a very nice question. So it really depends on the changes that the RLD product has. So it may be so the best way to address this is so sometimes it is you don't have to repeat any B studies if the change is very, very minor. And sometimes you need to repeat the in vitro B studies or sometimes in vitro and in vivo study. It really depended on the changes. And based on the data you provided, additional B studies may be recommended. And again, we encourage you, once you identify that the RLD product has changed, you contact the agency as soon as possible. And you can propose, you can delineate what changes the RLD have, and also propose the studies that you think may address these changes. And also, if you think no changes need, no B study need to be conducted, we recommend you to also provide justifications for that. And we encourage you to contact us as soon as possible during the pre-ANDA phase through the control correspondence or product development meetings, or if you identify this after your ANDA has been submitted, you can contact the regulatory project manager regarding how to proceed in this case. Thank you.
Thank you for responding to those questions. Moving on to our next panelist. A few questions came in for Dr. Elizabeth Bielski, and here's the first question for Dr. Bielski. Will the B studies recommend change in the case where an approved MDI's generic propellant must be changed to a new and quote green propellant? Would this depend on whether the RLD has adopted a new green propellant, or said another way, should the generic sponsor plan to adopt green propellants be without regard to the RLD?
Thank you for that question. That's a wonderful question and a really good hot topic currently in the space. I'd say right now, as it currently stands, a lot of the green propellants, we would require safety data in order to assess them. So until a new drug product comes onto the market, we really have no pathway to approve generic versions. However, once we do get that safety data from the new propellants and a new drug product is approved using that type of propellant, then we may be open to really looking towards approvals of generic products. But we encourage folks to actually start adopting their bioequivalence pathway, even if the RLD doesn't necessarily transition. So it won't necessarily depend on the RLD. We look at both pathways, and if at that point We are open to kind of looking at the product-specific guidances for those products. So the bioequivalence studies we may recommend will most likely follow those, but we may also ask for additional data or information at that time, depending on if the generic, what the generic product of interest is, what the RLD is, and all the specifics. So at that point in time, I think we highly encourage folks who are interested in this area to come with us through our product development meeting process through the pre-ANDA pathway to have those discussions. Thank you.
Thank you for responding to that question. The next question for Dr. Bielski is the following. How does FDA determine which products to target for GDUFA-funded research to generate PSGs? Is it based on demand or interest from the generics industry, pricing concerns from payers and patients, or lack of competition based on Orange Book listings?
Thank you for that question. That's a really great question. And I think the way we can address this is we really look at all of those factors combined. I'd say first we consider the complexity. So if the generic product is concerned to be complex, we focus our priorities towards those complex generics. Then we also take into consideration how much interaction we had with industry in terms of interest in developing those products. We also look at the lack of competition as well as what the marketplace looks like. So all these factors combined kind of lead or gear us towards which products or which PSGs, which type of research we will fund and use to help us develop those guidances. Thank you.
Thank you, Dr. Bielski. This is referring to a question, our next question for you, that was responded to by Dr. Zhen Zhu, and it's asking, Q1 and Q2, is it not mandated for these products? Is this a correct assumption?
Thanks for that follow-up question. And I would like to clarify, yes, our regulations don't mandate us or require us to have a Q1, Q2 formulation sameness for inhalation products. However, we do recommend them most of the time in a lot of our guidances for inhalation products. So again, it's not a requirement, but we do recommend it. And then, of course, as you've seen with the Budesonide, Formoterol, Fumarate, MDI case for that generic, as well as other cases, if you don't want to use a Q1, Q2 formulation sameness, you can come to us and we can discuss what additional data or information we may need for that product. Again, I think we did touch base on this earlier in our panel discussion. So if you do have questions about this, we are always welcome to hear your thoughts through our controlled correspondence pathway or our product development meeting. Thank you.
Thank you for responding to that question, Dr. Bielski. And we do have one really long question for you that just came in. I can repeat it if needed, and here is the question. A clinical endpoint bioequivalence study was used to establish bioequivalence for the recent generic approval of Budesonide and Formoterol fumarate dihydrate inhalation aerosol as recommended in the product-specific guidance. A few other PSGs for solution-based metered inhalation aerosol products provide recommendation for alternatives to the clinical endpoint studies. Can those possible solution MDI alternative approaches to the clinical endpoint study mentioned in those PSGs be applicable to development of generic budesonide and formoterol fumarate dihydrate inhalation solution, a suspension-based MDI product?
Thank you so much for that question. It's a really good one and probably complex one. I think the agency generally is open to alternative approaches to these cases. The PSGs that mention possible alternative in vitro, in vivo, and in silico methods can serve as a guide to building proposed alternative approach for a budesonide formoterol from our MDI case, even though those alternative approaches is not mentioned in the PSG. However, recommendations are product specific and are dependent on the dosage form and formulation. So other inhalation products like suspension-based MDIs and DPIs can have some influencing factors as well as additional factors that may add to their product complexity, thus additional factors may necessitate additional information and or other studies. Therefore, you should really evaluate the critical quality attributes of your proposed generic inhalation product and choose those alternative in vitro, in vivo, and or in silico methods that support characterization of such specific product interest. Also help maybe establish your in vitro and vivo correlations or help establish overall equivalence at the local site of action. In this case, it would be the lungs. The proposed studies should always work together to provide a comprehensive evaluation of local drug delivery for establishing equivalence. We really encourage companies interested in development of alternative approaches to what is recommended in the PSG for budesonide, formoterol, fumarate, MDIs, case or other suspension MDIs or DPIs. You come to the FDA with a pre-ANDA product development meeting with your proposed alternative approach. Within that meeting, we really want to see your scientific justifications, your critical questions of interest, and that will allow us to have that proper discussion to see how that alternative approach may fit for your specific product of interest. Thank you.
Thank you for responding to that group of questions. Moving back to our previous panelists, we do have a couple of questions that came in from Dr. Fang Yuan. And here is the first question for Dr. Yuan. Dr. Yuan, can you bracket?
Thank you. It's a good question. We have encountered that during pre-NDA meetings and control correspondence as well. So in general, for complex products, the orally inhaled drug products, meter dose inhaler or dry powder inhaler are complex products. We generally do not recommend bracketing study design. Thank you.
Thank you for responding to that question, Dr. Yuan. We do have another question that just came in for you. Which specified microorganisms does FDA think needs to be controlled in the drug substance specification for MDI DPI products?
Thank you. Thank you for the great question. So I recommend you to refer to USP General Chapter 1111, Microbiology Examination of Non-Sterile Products, Acceptance Criteria for Pharmaceutical Preparations and Substances for Pharmaceutical Use. There are detailed recommendations. Thank you.
Thank you for responding to that question. Now, moving on to our next panelist, we do have a question that came in for Dr. Andrew Klerman, and here's the question for Dr. Klerman. Why doesn't the U.S. legislate for or incentivize the take-up of generics and thus bring the branded prices down?
Great question. So here at the FDA, I can't necessarily speak specifically to legislation. However, I will say that the American public does already have a pretty robust cost savings from approved generic drugs. There was a study by the Association for Accessible Medicines back in 2020 that found that 90% of prescriptions in the US were for generic drugs, but that represented only 22% of the cost. However, in this case, for orally inhaled drug products, I think the issue is just lack of generic competition. But that is something we are certainly working very hard at and utilizing regulatory science to help push that field forward and bring more generic OIDPs to the marketplace. Thank you.
Thank you, Dr. Klerman, and just giving our audience a moment. If you haven't had a chance to enter your questions into the Q&A chat pod, please do so now. We're trying to take a look at a few more questions that are coming in to see if they're relevant to the presentations that have been provided thus far. All right, looks like we have a question that came in for Dr. Srinivas Bahara. And here is the question. How does the agency recommend sponsors for generic handle a change in formulation to a new green propellant from a quality perspective? And what issues should be considered?
That's a great question. It's in fact not a simple switch. There are a lot of challenges that one needs to be considered as we transition to low GWP propellants. Some of the considerations should be the drug solubility, potential for ostwald ripening for suspension based formulations, for example, um, and drug substance stability profiles. um, I could not I we may have to rethink about what are the studies that are required, but if you have any specific questions, we highly encourage you to request a pre-NDA development meeting. Thank you.
Okay, thank you for responding to that question. We're just surveying the remaining questions to see if there are any relevant to the presentations that have been provided thus far. Just a momentary pause for that to give our panelists a chance to review some more things that are coming in right now. So be right back in just a moment. Looks like we do have another question and it's for Dr. Andrew Klerman. And here's the question for Dr. Klerman. As a physician, what is your viewpoint regarding the therapeutic substitution of a lower cost product within the same category, such as ICS LABA?
Thank you for that question. Speaking clinically, I mean, I think I would be inclined to first and foremost provide the patient with a drug that I think is going to treat their condition to the best possible ability. But I think I would also consider what's the financial cost of that treatment. And I would certainly take that into consideration and work with the patient to provide the best kind of value for their treatment. And interestingly, I think obviously I work for the Office of Generic Drugs. I have a great deal of confidence in our evaluation of bioequivalence, but there was actually just a paper published in the Annals of Internal Medicine by Dr. William Feldman from Harvard, and he did a real world evidence analysis looking at the Advair Diskus brand name versus the Advair Diskus approved generic or Wixela Inhub, and compared a wide variety of patient outcomes between those two, and found they were essentially identical in terms of clinical outcomes. So I'd encourage you to look up that study. It's by Dr. William Feldman in the Annals of Internal Medicine, published last month. And I think studies like that should provide a great deal of additional confidence in the therapeutic equivalence of these products. So I would have no hesitation in prescribing these products. Thank you.
I want to thank all the panelists and the presenters for answering a numerous amount of questions that came in from our audience. We'll now enter into our morning break and return at 11:05 a.m. Eastern Time. Enjoy your break.